Now starting with the next topic, auxin transport. So I give you the brief idea that auxin transports from the shoot apex towards the root. Okay, we already discussed last day that if this is a coleoptile, okay, and auxins are synthesized here. So auxin transports from the shoot towards the root. Okay, they are transported from the shoot towards the root. So this is shoot, this is towards the root, which is base. Okay, so this mode of transport from the shoot tip to the base of the plant, this is known as basipetal transport. Okay, so auxin shows basipetal transport, also known as polar transport. So the term basipetal means towards the base. Okay, there is another term named as acropetal, which is opposite to the basipetal. The term acropetal means, so acro means what? This acro term means apex. Okay, so remember the word towards the. So if it's basipetal, it will be towards the what? It's basi, so towards the base. It's acro, so it is towards the apex. What kind of transport does auxin shows? Auxin shows basipetal transport. Okay, also known as polar transport because it is in the vertical direction. So see, so IAA, which is auxin, it moves from the apical to the basal end, known as basipetal transport. Now they transport only from the top to the base. Okay, that is why it is a unidirectional transport. Now this auxin, when it is transported, what is the role of the auxin? They helps in plant growth. Okay, and this is the only hormone which is transported polarly. Rest of the hormones can be transported laterally also. Okay, so this is the only hormone which is transported polarly. Now the transport of auxin from the shoot tip towards the base is an active process. It requires energy. Now this transport of auxin occurs through the phloem also, and a little amount. So if it's transported from shoot to root, if the auxin is transported from shoot to root, then the vascular system involved is phloem. If it is transported from root to shoot, then the vascular system involved is xylem. Okay, now this is very important to remember because question came from this topic. Okay, the one which I highlight very important, that you will give extra focus on because questions mainly come from those topics. Now we will start first. Before starting with the transport, we will have to first study about the transporters involved. So first transporter involved is AUX1. Okay, this AUX1 is a transporter, symporter, which is located on the plasma membrane and it helps in auxin influx. Okay, it helps in the entry of the auxin into the cell. Okay, and this is located in the apical portion of the cell. I will show you in the diagram where it is present exactly. Now pin is what? This helps in auxin efflux. So one helps in entry, pin helps in exit. AUX1 helps in the entry and pin helps in the exit of IAA. So exit transporters are present in the basal end and entry transporters are present in the apical end. Okay, so look here at the picture. See, if I will draw, you'll understand it more better. Okay, So if I draw here, this is the preceding cell. Okay, this is the cell where I'll show you all the reactions occurring, and this is the following cell. Okay, so what we see that, wait, so let me draw again. This is the preceding cell, the cell before. This is the another cell, and this is the next cell. Okay, so these red ones are Auxin transporters. Auxin transporters, we studied that they helps in Auxin entry. So they are the influx proteins. Okay, now these are the efflux proteins named as spin proteins. They helps in the exit of the IAA. Okay, so next if I change the color, so see, I'll denote IAA with green color. So see, in the preceding cell IAA was there. When inside the cell, IAA exists in the negative form. Okay, this form of IAA, which is charged, negatively charged, this is known as deprotonated form. Okay, and IAAH is the protonated form. Okay, the protonated form is the lipophilic molecule. Lipophilic means they can pass the membrane on their own without the requirement of any transporter and without the requirement of any energy. Okay, so the protonated form is the lipophilic form. So what will happen? IAA from the preceding cell, okay, will be released through the pin proteins. See, the pin proteins are here also, through which the IAA will exit. Okay, so through which IAA will exit the cell. So IAA will exit from this cell here, from this cell here. See, it is not entering directly into the next cell. It is coming out in the cell wall region. Okay, so I label this region with another color. So this region is known as apoplastic region. Okay, so IAA from the previous cell is released first in the apoplastic region. Okay, this apoplastic region have low pH. Remember, low pH means it has acidic pH. Okay, and this inside the cell, which is cytoplasm, has neutral pH. Okay, so what I said, that the cytoplasm has neutral pH and the apoplastic region has low pH. Okay, so IAA minus from the preceding cell here, this is one IAA, this is another IAA. So there are several molecules of IAA which will be released through the efflux proteins. What are efflux proteins? They are the pin proteins. The purple color one are the pin proteins. Through the pin proteins, they will be released in the apoplastic region. Okay. Now, when they are released in the apoplastic region, what will happen? They will enter into the next cell. Okay. Now, these IAA will enter into the next cell. Now, entry, the protein responsible for entry is Auxin. Okay, so these IAA will take entry into the cell, IAA minus. Okay, it has taken entry into the cell since it was said that this Auxin is a symporter. That means it is transporting another substance along with IAA in the same direction. So it transports 2 protons inside. Okay, so along with IAA, one molecule of IAA minus, they transport 2 hydrogen inside. Okay, so the role of Auxin we studied. Auxin is located on the plasma membrane. They helps in the influx or the entry of IAA. Okay, they are the symporter. They transports 1 molecule of IAA with 2 molecule of proton inside the cytoplasm. Okay, the role of Auxin is done. Now coming to the next transporter, pin. Now pin helps in efflux. So once all IAA are here, once IAA minus, and I'll write IAA with the same color. Okay, I'm not changing the color. So once IAA minus have been accumulated in the cell, what will happen? They'll be released through the pin proteins into the apoplast. They'll be released into the apoplast. They'll be released into the apoplast. Okay. Now the same process will happen as it was happened earlier. Okay. Now inside the cell, what happens? I said that there is 2 forms of IAA. So about the first form we discussed. Okay. It is released from the previous cell, comes out in the apoplast, then takes entry through Auxin inside the cell and is released again from this cell by pin. Okay. Now I also discussed the second form of IAA which exists. This is the protonated form. Now protonated form is lipophilic. That means they do not need any transporter. That is, they can take the entry without the help of any transporter. Okay, so this is IAAH. So what happens? This IAA minus which are released here, these IAA minus which are released here, they react with H plus. Where are they reacting with H plus? They are reacting with H plus in the apoplastic region and forming what? And forming IAAH. Okay, so these IAA minus is reacting with H plus in the apoplastic region and forming IAAH. Okay, now these IAAH can take the entry into the cell. Okay, so what happened? IAA minus reacted with H plus to form what? IAAH. Now this IAAH will take the entry into the cell without the requirement of any transporter. Okay, but once IAAH enters into the cell, they rapidly dissociate. IAAH, when it is there inside the cell, they do not stay in the protonated form. They dissociate into IAA minus and H plus. Okay, now if I summarize the whole pathway, what happened? The entry was done by Auxin, this red one. The exit was done by pin, which are the efflux proteins, efflux transporters. Now IAA from the previous cell comes out through the pin proteins because this is an efflux protein. While they are coming out from the pin protein, they first come out in the apoplast. Inside the apoplast, some of IAA minus combines with H plus to form IAAH. Here also I have shown and here also I have shown. This IAH is lipophilic. They can take the entry without any transporter. Once IAH has entered the cell, what will happen? They will dissociate. Okay, because inside the cell IAH cannot exist. Inside the cell they has to break up into IA minus and H plus. So this was some of the IA minus was converted to IAH. Some remained as IA minus, which are transported through the transporter, because the charged molecule cannot pass on their own. They need a transporter to get the entry into the cell. So in that case, IA minus is transported through the Auxin transporter or the Auxin symporter inside the cell. Okay, so the entry and exit is clear. Now if I go to the picture back, so look here. In this picture, see, these ones are the Auxin proteins. These are the Auxin proteins. So these are the Auxin proteins, these ones. Okay, and the purple one, which I'll color here, these ones are the pin proteins. Okay, they are the pin proteins. Now the ones which is transporting IAA, it is Auxin. Now this one is Auxin. Okay, the one which is releasing or helps in efflux are the pin proteins. Now see, this region is the apoplastic region. They are the apoplastic region. Okay, IA minus is when released, some remains in IA minus form and they move inside by the symporter, but some of them combines with H plus to form IAAH, which does not require any transporter and they can move inside on their own. So IAH has entered through the plasma membrane without any transporter and immediately got dissociated once they are inside the cytoplasm. I already explained. Okay, and look about the pH. The pH of the apoplast is acidic, which is 5.55 to 5.6. Okay, now the pH of symplast or the pH of the cytoplasm is 7. Clear? Now they need to maintain this pH all the time. Okay. So this I discussed, that negatively charged molecules cannot pass through the membrane, so they need the transporter. But the lipophilic molecule, which is a neutral molecule, it can cross the bilayer. Okay, already discussed that H plus Ia symporter, they co-transports 2 protons along with oxygen ion. So this OX1 is a kind of H plus IAA symporter. Okay, this is a kind of H plus IAA, or another name of OX1 is H plus IAA symporter. Okay, so it is transporting 2 protons together with 1 molecule of IAA. Now this transporter allows the greater oxygen accumulation than simple diffusion. So when IAH moves alone, it does not meet the requirement of the plant. Okay, the symporter which is involved, it transports more and more amount of oxygen to the next cell, which can meet the demand, but in this process energy is required. Okay, so in oxygen efflux, what I said, that the pH is 7.2 where? In the cytoplasm. So in the cytoplasm which has a pH of 7.2, in the cytoplasm which has a pH of 7.2, IAA cannot exist in IAH form. IAH readily dissociates into IA minus and H plus. Okay, they dissociate to other anionic form. Now, once IA minus is formed, they cannot release outside on their own. They need a transporter. Okay, They are impermeable because they are negatively charged. Membrane doesn't allow the charged particles to move freely. So they are impermeable, so they require transporter to take entry and exit into and from the cell. Okay. So it will accumulate in the cytosol until there is a signal to release it to the next cell. Okay, then they escape via auxin efflux carrier, for example PIN. Okay, now how was this PIN proteins identified first? Okay, so it was discovered first in Arabidopsis. There was a symptom in Arabidopsis which showed that when this PIN protein, when any protein was absent in Arabidopsis was once analyzed. What happened? They found that when a kind of protein was absent, okay, in Arabidopsis, the inflorescence became pin shaped. Okay, they found that when one particular kind of protein was absent in Arabidopsis, then their inflorescence became pin shaped. In the normal situation, the shape of the inflorescence is something else. But when that particular protein, which they didn't know the name, when that was absent, the inflorescence became pin shaped. So they named that particular protein as pin proteins. Okay. So again, question comes from here as well. So see, this is the more elaborate form of the auxin transport. So here what happens, IA enters the cell either passively in the undissociated form I discussed, either they enter in IAH form passively without any energy, or they enter in the negatively charged form by a transporter, secondary active transport. So this is an active transport. Okay, the second it's saying that the cell wall is maintained at an acidic pH by the activity of the plasma membrane. So what we saw that here, that the apoplastic region has a pH of 5 and the inside the pH is 7. But what we see that due to this process, H plus is entering into the cell. When auxin is transported inside through AUX1, hydrogen is entering in the cell. Okay, again through IAH, hydrogen is taking entry into the cell. So if more and more H plus will always enter inside the cell, the stage will come when there will be lack of H plus in the apoplastic region. When there will be lack of H plus in the apoplastic region, the pH will rise up and it will make a disbalance in the plant metabolism. So in that case, to maintain the level of H plus in the apoplast, there is another transporter which works. That transporter is named as H plus ATPase. What this H plus ATPase does, it releases the H plus out of the cytoplasm. What we discussed, that the two processes of entry, okay, the two processes of entry, if I summarize, the two processes of entry of IAA, in both the process, H plus is entering into the cell. See, they are entering through AUX1. Through AUX1, 2 H plus are entering in. Through IAAH also, H plus in. Okay, but there was no way we saw that the H plus is released out of the cell. So if H plus will not be released outside the cell, the pH will be disbalanced. They have to maintain the pH of this apoplastic region to be acidic, which is around 5 to 5.4. Okay, so to maintain the pH, more and more H plus should be there. How this H plus concentration will be maintained in the apoplast? So this is maintained by H plus ATPase, okay, which will release the H plus from the cytoplasm into the apoplast. Okay. Now, I discussed that the direction of auxin transport is from the apex to the base. Now, this method of or this process of transport towards the base is known as basipetal transport. Okay. Now, in the fourth case, the anions which accumulate here, they cannot exist because they are a charged one which cannot cross the plasma membrane. So, they need efflux carrier proteins named as spin proteins to be released from the basal end. Okay, so this is the apical end. Okay, this part is the apical end and this part is the basal end. Okay, and the transport is a longitudinal transport. And the source of this picture is Tej and Zeiger. Please follow that book to study it elaborately again. Now, inhibitors of auxin transport block auxin efflux. So, mainly the inhibitors are found that block auxin efflux than blocking auxin influx. Okay, so auxin efflux are mainly blocked by inhibitors than the influx. So, two inhibitors which are mostly found. The first is NPA. The full form is naphthyl-thalamic acid. Okay, pH is silent, naphthyl-thalamic acid, and the second is TIBA. Okay, now NPA and TIBA, they block the polar transport by blocking auxin efflux. Please remember, they block auxin efflux. Okay, but they do not block AUX1. That is, they cannot block auxin influx. Okay, now TIBA, now understanding about the role of each inhibitor, TIBA have a weak auxin activity. Okay, they are also transported polarly like auxin and they compete with the release of the auxin through the efflux carrier. I will show you. If this is the cell, if I first study about TIBA, TIBA have a weak auxin activity. So here what are protein? Here are the pin proteins. Okay, so if IAAs are here, if blue colored is IAA and these red colors are the inhibitors, red colors are TIBA. Okay, so TIBA are red colored. Okay, so what TIBA will do, TIBA have a weak auxin activity. TIBA will go and bind to the pin proteins so that IAA minus cannot release. So since they have a weak auxin activity, they go and bind to the pin and doesn't allow the auxin to bind to the pin proteins. Okay, in that way they inhibit the auxin efflux. Now second one, which is NPA. The full form is naphthyl-thalamic acid. What this does, now they do not have any weak auxin activity, they do not look like auxin, but what they do, it was found that NPA binds to any protein which is responsible for auxin efflux. Now they did not mention which protein they bind to. So if I consider on ourselves that pin proteins are the ones which helps in auxin efflux, so they bind with any proteins which helps in auxin efflux. So let's consider pin helps in auxin efflux. So they are binding with, say for example, pin proteins. So once this NPA is binding to any of the proteins which is responsible for auxin efflux, they are not allowing any efflux transporters to work. So to which any of the efflux proteins, when they will bind to, they will block the efflux transport. So if they bind with pin, they'll, say for example, they'll change the conformation of those pins to which they bind and will not allow the auxin to be released through those pins. Okay, in that way NPA inhibits. Okay, so TIBA has a weak auxin activity which competes with the auxin and does not allow the auxin to be released. They themselves occupy the site of the pin and does not allow the efflux of auxin. NPA binds to any proteins which is responsible in auxin efflux. So any proteins which is responsible in auxin efflux, NPA bind to those. Okay. Another inhibitor named as naphthoxyacetic acid, in short NOA. This is the inhibitor which blocks auxin uptake into the cells. The two we discussed, that is NPA and TBA, they block efflux. But NOA, which is naphthoxyacetic acid, it blocks auxin uptake. Okay. Now we discussed that there are two proteins, AUX and PIN. Okay, so AUX1 always stays on plasma membrane. Whether the auxin is transporting or not, they always stays on plasma membrane. So this is very important to know because question came from this concept also. Now PIN are the ones when the when there will be no transportation, when the pin are not in use, what will happen? The pin were also present on plasma membrane. So when, say for example, they are not required, what happens? They move inside the cell and become enclosed in the endocytic vesicle. Okay, they are compartmentalized in the endocytic vesicles and they are recycled back to the plasma membrane. Okay, they are compartmentalized into the endosomal vesicle or endocytic vesicle and they are recycled back to the plasma membrane. Okay, so AUX1 always stays on plasma membrane and PIN are recycled. Okay, when not in the requirement, they move inside the cell and become enclosed in the compartment, and once they are required again, they move back to the plasma membrane. Okay, this concept is again very important. So my motive of writing very important means questions came from these topics. Now the next, another efflux protein which was identified is known as ABCB protein. So like the PIN protein which was efflux protein, so we studied two efflux proteins now. One was PIN and the second we studied is ABCB. Okay. Now what are ABCB proteins? They are ATP binding cassets of family B. ABCB98 is used for the rootwards transport of auxin. Rootwards means towards the root. Okay. Now just remember ABCB is a efflux protein. Now one question if we solve, during development of embryos in plants, PIN proteins are involved in. So what is the role of PIN protein? If I give you few seconds to solve this question. They help in the establishment of auxin gradient. They help in the regulation of gene expression. They help in induction of programmed cell death. They help in induction of cell division. Okay, so it will be establishment of auxin gradient. So what happens? What do PIN do? So PIN are here. The concentration of IA minus is lot more inside. And it is very less here in the apoplastic region. In the apoplastic region they are very less. Okay, so they are moved from the higher concentration to the lower concentration along a gradient formed by what? PIN. Okay, so these IAA just a moment. So these IAA which are here in high amount and here they are in the low amount. So they along the concentration gradient formed by pin, they moved from the outside to the inside. Okay. So this pin proteins form the Auxin gradient. Okay.